Hello and welcome to Bio 138. And today what we're going to be taking a look at here is this concept that many of you guys have seen before, which happens to be the scientific method. And this, of course, term, when you take a look at it, is kind of unique. And part of the reason why it is unique is because back in about the 1960s into about the 1970s, we happen to have, of course, a lot of different technological advances. We had a big emphasis in the science area. And so our science publishers, those that make the textbooks and everything, especially in K through 12 education, were really sitting down and they were taking a look at, how science is done and, how do we write about this stuff and, what are we going to call it? Because nobody ever really sat down to document how this process is done. And so the textbook publishers actually came up with this idea, which we call today the scientific method, to kind of, if you will, simulate how research is conducted in the lab. And I would say for the most part, it's probably about 70% accurate with about, some 30% intricacies and differences that, again, you would not normally read about in a textbook. And if you actually saw these differences occur in a lab setting, some of you, if you just read books the whole time, would actually go ahead there and be like, well, that can't be the way it's done. But in reality, it really happens to be. So we're going to take a look today at the process of the scientific method. Okay? And so with that, what we're going to look at with the learning objectives is being able to list and explain the steps here to the scientific method. And then also the big idea right here, which happens to be that a hypothesis is going to be falsifiable. And then we'll talk about control groups, talk a little bit about an experimental group, and of course talk with that experimental group about what we call the experimental variables. And then finally, really hopefully, I wish we could actually spend some more time with this, but we don't have that time. But really talk here about this word right here, which happens to be facts. A lot of individuals out there really love this word and they go, oh, I just need to know the facts. I just need to know these because they're so important. When in reality, they really don't do a whole lot for science, and I'll be getting into that in a bit. Okay? So we're going to talk about facts, hypotheses, and laws that kind of come together to give us an understanding here within science. And as I mentioned here in previous lecture number 1, anytime that you happen to see this term right here, lecture, this is something exclusively in the video. And anytime that you see a page number, this is where you can look that information up within your textbook. Okay? So these happen again to be the 5 objectives right here that I'm going to be taking a look at here today with this PowerPoint. Now, as you take a look at this PowerPoint, I happen to have this image in front of you, and if you look at it for just a few moments, you're probably like, oh my gosh, what's going on with those frogs? And you can see right here, that this frog right here actually happens to have 3 hind legs, so you've got one here, a second, and then you have a third. And then if you take a look at this one, he's actually growing an additional hind leg right here off from the hip. And then if you look at this little guy right here, he doesn't even have any hind legs at all. And so if you notice right here, we happen to have some frog deformities that are going on, especially as it relates there to the limb formation. And so as we look here at the scientific method and everything that many people here have kind of come to know and understand, even though you can go to different textbooks and they'll kind of, show it to you differently, but every, again, book and every, scientific method is going to begin right here with some sort of observation, something out there that is going to go ahead here and really make you begin to wonder, like, what's going on. So when I, again, showed you this image up here previously, you're like, oh my gosh, what is that? And as you looked at that image, you're probably sitting there going, there's something unique here about these frogs, which then gives you this part right here, which happens to be the question. And the question right here, these are very general types of things. And when you take a look here at this question is, basically what is causing the frog deformities? We could easily put that into a question format. And if you notice when I mention that term, what is causing the frog deformities. Notice I didn't go ahead and I didn't put in a possible explanation. I ended up just going ahead here and I put in something that is very general. Because again, looking at these frogs and everything, you can see that there's a lot of different, formations here that are going on. So it's really hard right now to go ahead and nail one type of thing. But, looking at it right now, we can see that there's something that's causing these frog deformities. Well, ultimately, if you're really trying to look at what is going to be a possible explanation, and notice how I said what is going to be a possible explanation, it's important to understand that sometimes in life there does happen to be more than one right answer. And that's the unique thing right here as we take a look at these frogs and their deformities, is that, you're looking at these frogs, you're probably looking at them in the environment, and you could start sitting there going ahead and saying, well, there must be multiple things here that are causing this. And maybe there are, maybe they're not. So there's many ways that you can go about answering that question. And what your hypothesis happens to be when you look at it in general, a hypothesis, what you're going to be doing is you're going to be going back here into the environment and you're going to be looking at, the habitat of the frogs and everything and seeing, can you take this and can you actually take the conditions here and really break them down and try to see, hey, these could be a possible explanation for that potential question that you've answered or that you've asked previously. So again, with that question, if you're wondering what is causing these deformities, your hypotheses for that could be potentially something as simple as, fertilizers. Could be something as simple as, let's say, some sort of agricultural pesticide or a pesticide that a, homeowner may be spraying. Maybe there's a nuclear plant in the area. Maybe there happens to be some sort of parasite that lives off the frogs that is in the area. Maybe there happens to be some sort of chemical spill, or maybe it's temperature related. And so when you look at our hypotheses, there's numerous different answers here for that question. That's really what our hypothesis looks at, is it's looking at a unique way to go ahead here and kind of answer that question. And so with that, hypotheses and everything, we're looking at a plausible explanation to that question. And now this becomes a really important concept that applies to all science, which happens to be that when we actually write down the hypotheses, it has to be written in a manner where it can be falsifiable. And what I mean by falsifiable is that you can actually go ahead and be inaccurate with that hypothesis, that another researcher could come along and actually prove you wrong, even if your data shows that you happen to be accurate. So this is kind of a tricky area here within the whole aspect of science and everything, because as a layperson, since this is a non-majors course, many of you, again, this is your last science course, and you have to understand this, that as you take a look at science and you look at these hypotheses, since they are falsifiable, there are ways to go ahead and to sow discord, and ways to go ahead and to put in things, if you will, that normally would get people to stray away from a really good potential hypothesis. And part of the reason why your hypothesis has to be falsifiable, you can use the example right there of a fortune cookie. You go ahead, you crack open a fortune cookie and you read it, and the fortune cookie says something to the extent of, you will accumulate wealth in the future. Well, taking a look at that, what you might consider a hypothesis that you're going to accumulate wealth in the future, it's very general, it's very vague, and there's no outliers that set any sort of limitation to the boundaries. And so right now when I record this lecture and everything, in a few days it's going to be Friday, and it's going to be direct deposit day, if you will. I'm actually going to get paid, and so thus I've accumulated wealth. Well, if that's my fortune cookie right now has been again, proven, if you will, to be 100% accurate. I came across wealth, and that's just, again, something that occurs every 2 weeks with the joys of being and having direct deposits. So we always want to have these hypotheses that are falsifiable. With that hypothesis, we also want to go ahead here, and before we even write out a potential solution, a potential answer here for the question, we also want to make a prediction about what we think is going to happen here. And with that prediction, we want to go ahead and try to predict, like, again, let's say hypothetically we look in the environment and we see that there happens to be some sort of fertilizer runoff. So our hypothesis is that fertilizer runoff is causing the frog deformities. Well, the next thing we want to really do here is we want to make a prediction, like, is this something that's going to predict, influence, if you will, 10 or 15% of the population? We actually want to have some sort of numeric number that is going to be kind of a standard right here, something that we can kind of start doing some comparisons to. And so again, we're going to kind of go out on a limb and say, hey, maybe let's say that the rate of deformities will increase by 10% again if there happens to be some sort of fertilizer runoff, again, that's occurring from the local environment, from people's lawns and everything when they fertilize. So then the next thing we would go ahead and do is we would actually set up a test. And when we set up a test, normally what's going to happen is we're going to have what we call a control group. And with the control group, this is going to receive no treatment. And in many cases, when you take a look at a control group, they do receive what's called a placebo, which happens to be kind of like a false treatment. So let's say that you're testing out like some sort of new medication that's going to lower cholesterol. Well, the control group, the placebo, receives some sort of inert. And when I say inert, that would be a chemical that does not react in the body. So it would just break down and do nothing to the body. And so the person would actually take the placebo, and they would think they are actually getting the treatment, but in reality, they're not getting the treatment, they're just getting this medication. And the medication, it's not even medication, it's totally an inert molecule. And so with that, they would take that, and that would give the experimenter a group to do some comparisons. And the reason why this group is there to do comparisons is because, again, this group, as you see it, did not receive the treatment, so you know what your baseline is going to be here. Now, I will say that there does happen to be what is known as the placebo effect, that sometimes when people perceive they're getting the treatment, their condition actually improves, that there's like some sort of like psychological thing that goes on in the brain, especially when you see diets. Like if people all of a sudden take a placebo, sometimes they lose more weight than what you would expect because they're just kind of now watching their weight. They're thinking about what they need to bring into their body and everything because they're receiving what they think is the treatment, but it's really not the treatment. They're getting the placebo, and so thus they're losing weight, but it's not because of this inert agent that they're getting. So that's the control group. The other thing, too, that you're going to set up is going to be known as the experimental group. And the experimental group, they're going to get the treatment. So let's say that I was going back here to my frogs, and so with that, I ended up going ahead and I just sprayed these with just some water, right? So the frogs just get sprayed with some water. In my treatment group, let's say that I go ahead and I spray some frogs here with 1 part per million, so 1 ppm, of a common household fertilizer. Now here's the other thing too that I can do with my experimental groups in my test. I can only test, so you only test one variable. And so as I said, my variable here is fertilizer, right? So that's what my variable is. Well, I can run several experimental groups. And what I mean by I can run several experimental groups I can have experimental group 1 right here, and I'm going to put a little E right here for experimental group 2. They're going to get the same treatment. And let's say I increase the dosage to 2 parts per million. And then, of course, I can have an experimental group number 3 that's going to get the same treatment, the exact same fertilizer. And let's say again that I'm going to give this group over here 3 parts per million. The important thing again is that I'm testing one variable, and that variable is fertilizer. So I didn't go ahead and I didn't test fertilizer with changes in temperature, because that would be too many variables. I wouldn't know, is it the fertilizer, is it the temperature that's actually causing some of these influences within these frogs and everything. So I can have, again, several experimental groups, but I'm not going to go beyond just having one variable. The other thing, too, when you set up these groups up above here, you always want to go ahead and set these groups up so that way they're always going to be the same setup. So I'm going to write that down here so you have, again, I have a little more white room down here, but you want to have the same setup. And what I mean by that is the control group, the experimental groups right here, all 4 of these groups. Let's say again that you have a species of frogs. Well, with your species of frogs, let's say that these happen to be, I don't know, let's just say that they just happen to be like a green tree frog. All the species for all these groups, the control, the experimentals, they would all be green tree frogs. The other thing you'd want to look at is the sex ratio. You'd want to go ahead and probably have the same ratio of male to female, or you could potentially just have all males, or you could even just have all females. But whatever you do, you want them to be the same, okay? Another part here happens to be the age, if you will. So with these frogs and everything, they develop from eggs. So you would want the eggs to be of the same age. So with that, again, I have age and eggs right here. So, since these limbs are something that develop inside of the egg and everything, you would want those eggs to be at the same age. So are they 1 day old when you start the experiment, or are they 5 days old? You would again want every single part of that to be the same. All right. And then the other thing that we want to try to do here is we want to try to get rid of some bias. So looking at the control groups and looking at the placebo groups, one of the ways that you can make this, again, eliminate bias is by having what is known as a blind study. So what would actually happen is one researcher would go ahead and basically erase the names here off from the placebo and off from these experimental groups. And then what they would do is they would use some sort of code, so that way the researcher would be the only one that would know what is actually going on to what group. Now, another way you can do that is with what's called a double-blind study, where you happen to have kind of a group A person. So you have person A, and person A, what they do is they remove all the labels off from the devices, and then they give that to person B, and the labels are there as far as, not telling you which one's the control, which one's the experimental group. You just would have like some sort of numerical identification that person A is not sharing here to person B. What person B then ends up doing is they take that numerical system and they change it completely around, so that way they can go ahead and they can run the experiment. And this is what is known as a double-blind study, because again, when you take a look at this again, person A is setting up person B, and person B doesn't know that, and person B changes everything around, and so thus the bias has been eliminated. Okay? Now, back to again this idea of the controlling experimental group. When it comes time to collect your data, so when your experiment is done, you get some observations here, you write down some more notes and everything. Let's hypothetically say that you had a number, and this is something I forgot to mention. Typically, when we're looking at these groups here, the controls and the experimental groups, normally we want to have like a population size. So we want to have a population size that typically is above 20. And what being above 20 starts doing is it starts going ahead and it's going to flatten out some of the probability numbers. So that way the numbers are just a lot more easy to work with because they're not as drastic of a change if you have a group of 20. Let's say you flip a coin once and it comes out heads. Well, your data right there would say 100% of the time when you flip a coin, it's going to be heads. Well, we both know that if you flip a coin twice, it may be heads the first time, it may be tails the second, or you could have 2 heads and 2 tails. So if you flip a coin about 20 times, you eventually see that your probability, your chances of having a heads or tails, right, is about 50% because you have flipped that enough times. And this is something, again, like I said, I forgot to mention right here, is that all of these groups, you typically want to have a population number that's going to typically be above 20. You can be below 20, but probability kind of starts going against you a little bit, and with that, you do have to watch your statistics there. So again, you do have to be careful of that. So the next thing that we would go ahead and look at, like I said, we would collect our data. And so from that point forward, the next thing we would do is, let's say that, you've got the control group, and let's say we had a population size of 20. And so in the population, let's say that you had 3 frogs that received just water that had a deformity. So you had 3 out of 20. And so then you had experimental group number 1, and they had 4 out of 20 with 1 part per million. And then you had experimental group number 2, and they had 3 out of 20. And then let's say you had experimental group number 3, and they had 1 out of 20. The nice thing about this control group right here is it allows you to compare what has happened with these events right here that you changed the variable with. Well, if your control group is showing you 3 out of 20, and these experimental groups, you had a 4 out of 20, a 3 out of 20, and a 1 out of 20. If my, again, hypothesis is that fertilizer is the cause here of the deformities, most of you would go ahead and say, you know what, I don't think so, because these numbers all look close together. Now, the other thing here that we would end up doing when it talks about this additional observation is we would use some statistical, or we would use some stats right here to help us break these numbers down and compare them. And there's some different statistical analyses that are out there, but I don't want to go into them, because if I did, that literally could be days upon days about talking about different ways of statistical analysis. But we can see clearly here from the numbers there's no difference. Well, back to it, what's going to happen here is we're going to say that our test does not support the hypotheses. We're not going to say that we're wrong. A lot of students want to say, oh, we're wrong. It's not that you're wrong, it's just that right now, given the conditions that you have, you're just not supporting the hypotheses. There's a difference between being wrong and just not supporting something. Like, for example, there was a time when, there was a lot of people out there that happened to believe that if you went ahead and if you, had a diet that was low in fat, that you would go ahead and lose weight. Well, turns out that low-fat diets really were not working because you would substitute fat for sugar. And really, your test right here, it's not that you're wrong when you set that up. It's just that right now, the evidence isn't supporting it. And maybe if you, change that diet up when you start talking about, having a low-fat diet or having, again, a low-sugar diet and things of that nature, we have to look at it in some different ways before we end up finding a hypothesis that we support. So there's different ways to go ahead and answer these questions. And I think that's always going to be the most frustrating thing for many people to understand, is that science at that time is going to have one answer. And that answer can change as we get new pieces of data and as we look at things at a different light. You know, there was a time when people said eating eggs was bad for you. And so we tried not to eat as many eggs, and now people say, you know what, eating eggs is healthy. Or another one happened to be out there that you shouldn't go ahead and use butter. So we're using things like margarines and different types of oils and everything else. And it turns out, you know what, the butter's not actually as bad as something like the margarine happens to be. So there's a lot of data out there that has changed just because our understanding of cholesterol. You know, there was one time when we only understood cholesterol in general. We didn't understand what HDL happened to be and LDL, which happened to be high density, and then also low density. We didn't understand that at all. But now we get a better understanding. So thus, you know, this whole concept right here is going to always be changing back and forth. And that's important to understand. Okay? Anyways, getting back to my frog experiment here. If your test does not support the hypotheses, what do you need to go ahead and do? You need to come up with a whole new one. And so when you go ahead now, and let's say that you're going to come up with a new hypothesis here. So I'm just going to real quickly go over to my eraser and everything here. I said like fertilizer, right, was the big thing that ended up influencing this. And I ended up having, you know, all these, you know, 1 parts per millions and everything else here. So I'm just going to real quickly go ahead here and I'm going to erase that. Go back over here my pen. So let's say that it actually happens to be something like, not a fertilizer, but let's say it's Roundup. Roundup is an herbicide. So you do the same thing. You get like 1 part per million of Roundup. And let's say you do like 1.5 parts per million. And then you do 2 parts per million of Roundup. And you keep your placebo the same. Well, let's say you collect the same type of data here. Well, back to it. Is it Roundup that's causing this? Is it going to be fertilizer that's causing it? The answer is no. So you keep coming back to this and you keep rewriting this and you keep redoing this. The unique thing that we end up finding out, just to kind of let you know, is that in some cases, like with these frogs, one of the causes of deformities is what's known as a nematode. And a nematode happens to be it's a really small animal. And when that animal gets into, I can't spell today. When that animal gets inside of the eggs, it influences the life cycle there of the frogs. And so that's one tested explanation that actually explains why these deformities end up happening. There's actually a couple other explanations to explain frog deformities. So again, you know, when we start taking a look here at science, it's important to understand that based upon the situation and based upon the environment, that these frog deformities happen to not have any sort of one explanation. They do happen to have numerous explanations, and it all happens to, again, change based upon where you happen to be at. Okay, so as I was mentioning right there, these missing limbs and things of that nature was pretty big right there, and so that was a nematode. Another thing that causes some of these deficiencies is actually lack of nutrients in the frog's embryos when they're developing. And then there does happen to be some influences of some toxic chemicals that sometimes leak into the environment. So it's not one answer, there's actually 3 answers. And actually, there's even more than just 3, but just trying to simplify this here for today. I hope you kind of understand here that these frogs here are deformed, not from one thing, but actually several. And again, that's this whole idea of being falsifiable, that our hypotheses of this deformity is definitely falsifiable. We can have more than one type, again, of answer that does occur. Okay? So that's big, all right? Now, when we start taking a look here at science and things of that nature. A lot of people right here really think that your facts are like this really powerful thing. And actually, facts really happen to be kind of the least accurate. They're also the least predictive type of information that we could have in the field of science. So for example, right now, I am wearing a red sweatshirt. At this point now, go ahead and explain to me what you know about me if I'm wearing a red sweatshirt. Most of you are probably sitting there scratching your head saying, I can't explain anything. And that's my point. Some of you might say, well, he likes red. Well, honestly, I don't have a favorite color, so that wouldn't be a plausible explanation. I just have a red sweater on today, and that's it. It doesn't explain anything at all. But most people, when you go ahead and you listen to people, especially on the news, especially when you get opinion journalism is, not the reporting of the news. It's when people are adding their 2 cents, if you will, into it. And again, a fact is just that. It doesn't have any sort of power to explain. Now, if you go ahead and if you start putting a bunch of facts together and everything else, you may be able to go ahead here and develop some sort of hypotheses from that. But sometimes, again, that can be difficult. But as we go up here, this kind of pyramid that I have here, as you start making your way up, these things become more valid. They have more points to them. So again, a fact of just me wearing a red sweatshirt is just that. It's really low here on this whole idea of validity. But if I have something that does get tested, like a hypothesis that does get supported, that's going to have some sort of validity to it. Now, if I start taking all different hypotheses and I start putting them together, I can start developing what is known as a theory. And theories, again, happen to be supported by not just one hypothesis, but they actually get supported by many hypotheses. And so this data that comes in from all these different hypotheses to come together to make a theory makes the theory even more valid. And the great thing about theories is that theories can explain laws in science, and laws happen to kind of be these general statements that we end up getting that try to explain, like let's say something like the laws of thermodynamics in physics where, energy cannot be created or destroyed. Well, theories can explain the laws, and the laws can also help support the theories. And these two things go hand in hand, if you will. So in biology, what's kind of unique is we actually don't have many laws because we have a lot of different living organisms that find ways to adapt, if you will, to the laws. So when I say laws, I'm talking about like thermodynamics and everything. So in biology, we find a way to kind of adapt to these laws. So we don't make our own laws, but one of the laws that we have there happens to be that proteins are built on ribosomes. And so when I say that a protein is built on a ribosome, that is a law. And what is a ribosome? This is a structure inside of a cell, okay? And I'm not going to really get into cellular structure. That's going to be here for the future. But ribosomes are where you build proteins. And so with that, we happen to have theories here, and one of those theories happen to be Mendelian genetics. And with Mendelian genetics, we understand here that with genetics, that, a gene is the genetic information that is the code for making a protein. And so if you notice right here, if a gene is a code for making a protein, well, that helps to explain this law that the protein, again, is built on a ribosome. This is where we actually do this. And this law helps to explain this theory that these genes need ribosomes. So if you notice this kind of circle right here, that's ever so important, that the laws explain the theories and the theories explain the laws. The other thing to understand here too about my pyramid right here that I went ahead and I drew for you, when you start talking about the base here of this pyramid, the amount, this is going to be a really large number down here. And so, yeah, there's a lot of different facts here. And your hypotheses, there's going to be a lot less hypotheses than there are facts. And then, of course, with your hypotheses, when you go up to the top here, there's going to be, less theories than there are hypotheses. And so with that, there's even fewer laws than there are theories. And so this is kind of the way our knowledge ends up going. But when you get up to a law is the most valid thing that we have. The law explains so much of biology, if you will, or so much of the area that you're studying. And again, it's built upon other theories, and so they are there to support each other. So it's kind of unique when you start taking a look at it. And like I said, these little things right here, these little facts, they're really not as important as what the media, if you will, happens to describe to you. Okay? Now, if they describe some laws, great. If they describe some theories, great. But remember, they have to be valid ones. And that's another thing, too, that we run into with media. And it doesn't matter, where you get your media from. Because, again, the media will sometimes use the terms theories and hypotheses very differently. And sometimes they'll even use them concurrently, meaning that they're the same, but in reality, they're really not. So, anyways, this is the last slide, if you will, for Bio 138. So that's what I had here today for you. And this is what we all covered here today in this PowerPoint. So if you have any questions for me, great, shoot them to me. Otherwise, I hope that you have a great day. All right? If you have any questions, feel free to reach out to me. Otherwise, I'll talk to you later. Bye-bye.